Thank you very much. I'm honored for this invitation to share with you the findings of our research, which I've conducted with the expert direction of Dr. Erdman Rapp. Yes, I will go to the agenda. Today we have the introduction on the samples I've analyzed, the problem we want to acknowledge, and the method I developed for an in-depth glycoproteomic analysis, and how might this benefit your research. An application on IgA, which can exemplify well the benefit of this workflow, and the conclusion. Well, human blood plasma, as many of you already know, is a very complex sample. When we extract it by centrifugation, we will see this yellowish fluid, which contains hundreds of proteins. However, high abundant proteins represent the 95% of the total amount of proteins. So in this plot, I show you the protein rank and a large concentration range of 10 orders of magnitude. And our aim is to reach the low abundant proteins because they might be associated more specifically with tissue leakage proteins and therefore become potential biomarkers. Our hypothesis can be summarized with this little plot, where in yellow you see the circle area corresponding to the high abundant proteins, which are important but are not interesting for us in order to reach the potential biomarkers. We want to go to the center, to the low abundant proteins, and even better, to the rare glycoforms. And these are really the minor abundant ones. But if we have success in robustly identify them, we can offer a chance to explore the association of these glycoforms in different contexts, diseases, and conditions. So let's have a look to the sample preparation workflow. We develop a fractionation composed by two steps. The first, depletion of the top 14 high abundant proteins, and then a protein fractionation by molecular size using gel-free system, followed by triptych digestion, glycopeptide enrichment. We generate two elution fractions, which are measured by high-resolution mass spectrometry using two fragmentation energies, which are complementary. The second one, HCV low, can add structural information on the description of the N-glycan antennas. And these measurements are using intact glycopeptides. We don't do any chemical modifications. In summary, the first article, which I published in Engineering, describes well the method developed, which is composed by the sample preparation workflow and the data analysis workflow, which has some updates coming soon. But today I'm going to show you the gains of the workflow. As I mentioned, with the direct analysis of blood plasma, in this plot, you observed in blue the proteins that could be identified. They are mostly in the middle or high abundant range. With our workflow, the dots in red, we can go deeper and observe proteins in concentrations of nanograms and even one very, very low abundance. But this is not all. We can also observe better the microheterogeneity description of middle or high abundant proteins. For example, zinc alpha-2 glycoprotein. In this plot, you observed that with the direct analysis of blood plasma, we observe only 2 glycosylation sites and only 1 glycan. But when we use our workflow, we increase significantly the number of diverse N-glycans that we can robustly observe, and even more glycosylation sites. So how this workflow can benefit your research? You might wish to use this workflow if you are dealing with characterizing novel glycans, if you are working with mass spectrometry, of course, or if you are mapping post-translational modifications and you want to rapidly screen specific modifications in proteins. And I want to show you an example. Well, there were reported since 20 years ago sulfated carbohydrates derived from IgA from different species, mouse, rabbit. And in the latest 3 years, we have also observed glycomic analysis that reports acetylated and sulfated N-glycans from released human IgA N-glycans. Especially our group through the effort of Samantha Chahik, she developed a glycomic workflow that successfully characterized one of these N-glycans, the one highlighted in red, and with the use of a specific sulfatase discovered by Lea Chusel, they could confirm that actually IgA, human IgA, have hexanak sulfated N-glycans. However, when we talk about IgA, it's the second most abundant immunoglobulin in blood plasma. We are talking about two subclasses, IgA1 and IgA2, and I want to only briefly talk about this. We observe that IgA1 and 2 have homolog sites, highlighted here in orange, and IgA2 have additional 3 sites. So it's very interesting because glycoprotein's functions are depending on glycosylation and also on the position the glycosylation is. So it's really significant to know where the sulfated glycan come from, and this is why I apply the workflow. So first of all, we want to know if you collected mass spectrometry data, you want to confirm that you collected also sulfated glycopeptides, and we can rapidly screen for MS2 spectra containing these rare oxonium ions. And in this scheme on the left, I show you a symbol of a spectra that have different fragment ions that are in gray, typically from glycan building blocks of glycans. But in orange, you observe signals from sulfated oxonium ions. And what you want to observe are the signals in which the common oxonium ions align to the sulfated oxonium ions. And there you can find your special spectra. Later, we focus on the spectra containing rare oxonium ions, and we first try to identify the peptide moiety, but only the fragment ion to be able to calculate the N-glycan mass and the composition. It's very important to start the other way around, starting understanding the glycan and not the peptide. This will facilitate the glycoproteomic search. Later, we can also apply a wildcard search once you have a imagination of which compositions can be included in your data. In our case, which sulfated N-glycans and acetylated N-glycans were there. And then we can conduct glycoproteomic search that explains perfectly the peptide and the glycan. And this is how we could answer that this very interesting sulfated, hexanak sulfated glycomic glycan, well described by glycomic analysis, is attached to the tail piece of IgA. Actually, we could compare here in the bottom the non-sulfated N-glycan compared to the sulfated one, and we can differentiate perfectly the oxonium ions and the Y ions that confirms the presence of sulfation on the hexanak and on the antenna. We could also even describe better the peptide and other peptide variants. And in this way, I could confirm the glycan compositions reported by glycomic analysis, but surprisingly, we could also observe more compositions, and even one with 2 sulfations or acetylation plus sulfation. And I could map all of these compositions, of course, on every glycosylation site of IgA. And I can also mention that we studied 2 samples from 2 vendors. So this IgA is extracted from human blood plasma from 2 different sources. And we could confirm that sulfated N-glycans were present consistently in both samples on the homolog sites of IgA subclasses. On the contrary, O-acetylated N-glycans were mostly on the IgA2 subclass and were only confirmed on the sample from provider 1. And interestingly, the ratio IgA1 and IgA2 was different between both samples. Sample 1 was richer in IgA2, which is actually the correct ratio in blood plasma. Normally, there is 10 times more IgA1 than IgA2. And in this case, it could be interesting to know whether this observation of O-acetylated glycan only in sample 1 would be explained because IgA2 probably is richer in O-acetylated glycans than IgA1. this is a question that needs further research. Well, we could also quantify the sulfated and acetylated glycans in context of the total glycoprofile and site-specific microheterogeneity. Here in red, you observe the glycans associated to the TLPs, and in blue, the ones associated to the CH2 domain. And in this yellow area, we observe sulfated and glycans, and you observe the most predominant one is the one observed by glycomic analysis and is abundant in both sample 1 and sample 2, even though sample 1 and sample 2 have different amounts or levels of these glycans. Similarly, we could also quantify oacetylated glycans. However, only 2 of them were within the limit of quantification. And we could only quantify them at the TLPs of sample 1. Even we could identify contaminant proteins. Of course, there is a purification process, and the vendor provides IgA at certain purity, 99%, 95% sometimes. And surprisingly, this workflow was potent enough to differentiate glycopeptides containing glucuronidation and sulfation on the glucuronic acid, similar to the HNK1 glycoepitope, typical of brain glycans. And we could also identify sulfated glycans on other glycoproteins. In conclusion, sulfated and no-acetylated glycans exist primarily at the IgA TLPs, and between these 2 samples, there were significant difference in the identification of oacetylated glycans. Our new glycoproteomic method allows you to identify very low abundant rare glycans, expand the elucidation of IgA microheterogeneity, and sort out N-glycans derived from contaminant proteins. I want to also show the summary of the in-depth description of blood plasma proteins. When you do the direct analysis of blood plasma, you will see the tip of the iceberg and the typical glycans. But with our workflow, we were able to see lower abundant proteins, but also rare glycans, not only sulfated and glucuronidated ones, but also others containing very rare masses, reported for the first time by our research, which needs to be, of course, further investigated. Well, this is the outcome of this research. If you are interested in applying or using this method, please, you could have a look to have more details. I want to acknowledge the support of my former supervisor, Dr. Rapp, and for future Glycera and PI, and your attention, of course. And this is only an invitation to a conference for the next year. It's very early, but if you are interested, please have a look. Thank you very much.